It's a great pleasure to welcome KELIN XIA today. KELIN comes from Singapore, Nanyang Technological University, has a very broad interest in machine learning, AI, as applied to a wide variety of problems. And you're going to talk to us today about mathematical AI for molecular sciences. So over to you, KELIN.
Thanks, Ben, and thanks to the organizer for the invitation. So today I'm going to mention a little bit about how to combine mathematics with AI and do some application in the molecular science. So AI for science and AI-based molecular data analysis has been, a hot research topic for in the recent times. And one of the milestone works in this area is the AlphaFold models that was developed by the Google DeepMind team, which should not be too far away from the institute here, right? And what this AlphaFold did is actually translate the gene sequence information, the A, T, G, C, this kind of sequential information, which are comparably easy and cheap to obtain into the 3D structure information, which are very important to study the dynamic, the function, the property of the molecules. And these AlphaFold models, in particular AlphaFold 2, has done a fantastic job. He has achieved accuracy around 90, which is comparable with the high resolution experimental data. So this work was regarded as the breakthrough of the 2021, and along the work, a lot of the AI models has been developed. I don't need to convince you how powerful and what are the impact this AI has for a lot of the science and the topic. Well, as a mathematician, we're trying to say, well, we have so many nice AI models. What are the fundamental challenges and problems all these AI methods faced? We believe we can understand it from two basic areas. First one is this data representation. So what are these? Essentially speaking is that when we have a data, how we translate it into a suitable mathematical model. It can be geometrical model, algebraic model, or topological model. You have to present a suitable representation for the data. The second one is data feature learning. So essentially how you extract the most relevant feature so that it can be directly related to your functions. So that's the feature learning. Let's go to the real molecular part. So in the molecular data analysis, the same business. First, you need to find out what are the potential suitable representations. There are many ways to do that. If you are familiar with the sequential information, then you can use, gene sequence, amino acid sequence, sometimes use a smell sequence. So this provides you a way to represent the data. The other thing is that if you think I'm now interested in the 3D structure, the topological information, then you see the most widely used representation for molecular, which is this covalent bond-based graph representation. Essentially, each of the node here is an atom, and then the nodes are connected with each other through the covalent bond, which are very strong interactions among them. And if you say, I'm interested in geometric information, then you can propose some molecular surface representations. Here is one of them. What's going on is that each of the atom here, you associate with something called van der Waals balls. And then the outside of this van der Waals balls will give you a certain surface representation. However, you see this one, you have a lot of unsmooth regions. So what people usually do is that they have a water molecule goes around this surface, and then the trajectory of this water molecules gives you a little bit of smooth representation of the surface. So there are many other different ways to do that, but essentially they can provide you a geometric representations. But you say, oh, I want to go a little bit deeper and try some functions. And a lot of people study the quantum mechanisms, density function theories, and then you end up in a function representation of the things. If you study, deep learning, there are something called a 3D CNN, essentially represent as the volumetric data and then do the learnings. So here you see the same molecular, you can have different representations, and based on different representation, you can play the different games. There are actually two major types of games you can play. First one is called machine learning. So in the machine learning, what you usually input is the handcraft features. So these are the features you obtained by using some low knowledge. So we will go to that immediately. And then the other way is called a deep learning. So deep learning, you say, oh, I don't know all these rules. I just have a bunch of data. I'm very rich, so I buy a lot of CPUs and then throw the data in. You have this end-to-end learning. So essentially, in the process, they also learn some features, but this feature is learned through the fancy and complicated architectures. So here, the handcrafted features or the feature learning are actually has been used even before this machine learning business, something called a QSAC model, quantitative structure activity relation or quantitative structure property relations. So if you go to this model, you will see that people try to understand the molecular properties through studying their structure properties. So there's a lot of what they call a descriptor here, 0D, 1D, 2D, 3D, 4D. 4D is that you have the trajectory. So I don't go to the detail, but roughly speaking, all these things are structure representations. So why they focus so much in structures? They want to study function, right? But why they jump into the structures? This is because there's something called a structure-function relationship. So if you understand the structure well, you can be able to guess or understand the functions. One of the simple examples is this guy here. You see that this is a protein, and the most important characteristic you can see is that there's a hole in the middle. Right? Then you immediately wondering why there'll be a hole, right? The reason is that they have a hole because this called ion channel is located in the cell membrane, and this hole here will facilitate the ions transfer from extracellular to the intracellular. And then you have something called a volt molecular here. This is a bottle-like structure, and then the inside is hollow. So you imagine what this kind of protein can do. So essentially what's going on is that some part here or here may be open, and then the ions or some small molecules can come in. And then this bottle just carry these ions or small molecules through the bodies. So again, by just checking this simple structure, you already be able to guess some of the functions. So this, end up in a lot of these molecular descriptor fingerprint representations. And as a mathematician, again, when we see these thousands of descriptors, we immediately wondering, it is possible some descriptors are more fundamental, more intrinsic, and maybe say more about their structural properties. So this land up into the TDA business. Ruben give and Omar give a fantastic talk this morning about the TDA. So here I just approach the TDA from the user practical point of view, okay? So TDA is powerful because of three major properties we believe from the practical point of view. First one is high order representations. I don't need to say to the audience here, everybody knows this quite well. The other one is the topological invariant. Okay, why topological invariant is important? Because from the AI point of view, the key process, what they do is learn the information from one system and then transfer into the testing or the something you want to predict. So in this way, when you are transferring the information from one system to another, you hope the information you learned is more fundamental, more intrinsic, so that it has a better transferabilities. If you are only learn the very superficial properties, it may work for this system, but when you transfer to other systems, the generalizability may not be that well. However, if you are using this topological invariant, you immediately characterize the most intrinsic properties, and you immediately have better generalizabilities and transferabilities. Another thing is the multi-scaled representation. So the filtration process is a very important process. But from the physical point of view, why this filtration process is important? Because it naturally induces this multi-scaled representation. Essentially, with these growing balls, you will have a series of simplicial complexes. And this simplicial complex characterize the interactions and the various different scales. Imagine you have a high resolution microscope. Then you can see each of the points very clearly, right? But when your microscope's resolution get lower and lower, your image get blurry and more blurry, right? Then you begin to see certain clusters. And in the end, your resolution is very bad. Then you see all the things jammed together. So that means you are seeing the things in different scale when you are introducing this filtration process. More practically, what are these barcode or the result from TDA really gives us? So we can look at this simple example, this free carbon-16. So it's a cage structure made of 16 carbon atoms. It forms this kind of football-like shape. There are 12 this pentagon ring and 20 this hexagon rings. So the barcodes here are very regular. So what you can see is that here we use the Tor-algebra complex. The x-axis is the filtration parameter. So you can see that first in the Betty 0, there are actually exactly 60 bars, and the bars can be divided into two types. This one is slightly shorter. It's about 1.38 angstrom. This is exactly the carbon-carbon double bond. Because the double bonds are stronger, so their distance between the two atoms are shorter. So this one corresponds to the double bond. And then this one here is about 1.42 angstrom. Angstrom is 0.1 nanometer, is 10 to the negative 10 meter, okay? So this one corresponds to the single carbon-carbon single bond, because the single bond is comparatively weaker, so it's slightly larger. And then you have the short bars here corresponding exactly to the pentagon, and then the slightly longer one bars here corresponding to the hexagon. And then you have a very long bar here corresponding to the Betty 2 bars. So essentially, if your data is coming from real experimental result, then you should be able to decipher the meaning of each bars, and there are no room for noise in certain sense. And we can use these things to analyze some real biomolecular data. So one thing we can study is this DNA structure. We know that DNA is made of this A, T, G, and C nucleic acid, right? So if you go to the base part, you will see that all these A, T, G, C have this pentagon and hexagon ring structures. And this pentagon and hexagon ring will appear in the earlier stage of filtration, like the ones here. So by checking the bars, you can pinpoint to each type of the nucleic acid in the systems. And more interestingly is that you will see some bars here in the later stage of filtration. They actually tell you more global information. So here you see that if you have two pairs of this nucleic acid, they form a certain cycle. And then you have minor and major growth, they all contribute a cycle here. So essentially, with this kind of understanding, you can use a barcode like a fingerprint to pinpoint each structures. So with this understanding, we are trying to propose, there's also many other nice works along the line to use TDA to extract features from TDA and then combined with learning models. The general flow lies like this. You have data, you try to build up the suitable simplicial complex. From there, you extract or you learn the bars or persistent barcode or diagram, and then you select features and then throw into the either machine learning or deep learning models. So along this line, my former advisor and the collaborator Goei has done fantastic works. So he has used the TDA-based learning models in drug design. So there are many different tasks in drug design. Among them is this binding affinity prediction, and also this is binding affinity, roughly speaking, is how the drugs interact with the proteins that usually cause the diseases. So later I will also mention about that. And then they have this log P, log S study. Essentially, it tells you how the drug can be easily solved into water or solved into lipid bilayer. So you see that the TDA models not only working, but consistently deliver the better result than all the existing models in this area. And another even amazing result is in this grand challenging. So what's going on here is that in this grand challenging, the organizers, they have some experimental data that has not been disclosed to the outside people. Actually, one of the organizers is from the PVB data bank. This is very large structured data bank that are widely used by the biology people. And what you want to do, what the participants want to do is that they try to use their machine learning models. They don't care what model you are. You use MD, DFT, machine learning, deep learning, whatever you want, right? And then you just compete and then see if you can give a better prediction of the experimental result. And what TDA did, or Goei's group did, is that actually an earlier competition, it's just okay, get some medals. But in the later competition, you can see that TDA models are systematically deliver better result than existing models. So their group also did fantastic work in the COVID mutation analysis. Actually, in 2020 June, they began to post a paper online, say that there are two residues, 452 and 501, may result in the prevailing of the variations. So give you a little background. So this SARS or virus, if you count all the amino acids, it's in terms of thousands of amino acids. Here, they only find two of them. They said it's highly possible to result in a lot of the variations. And then immediately later, the bad thing happened. You have beta, gamma, delta, all this kind of mutation comes out. And then you check out these two residues are appearing in all of them. So this shows you the great power of the AI, the TDA model combined with AI can do in this mutation analysis. And another test case is that in 2022, they proposed a model to say that this BA4 and BA5 will become the dominant variation. And this is the time that this BA4 and BA5 just appear. And then later it become the dominant one as they predict. Shows again the great performance of the TDA combined with the learning models can do. So motivated by this great success, we do some works. One thing we started is this persistent spectrum. So we have the spectrum graph, from Shuangcha and Yao's work, and then spectrum spatial complex. Jürgen did a lot of nice work there, and then spectrum hypergraph. So what we did is trying to study the change of the spectrum information during the filtration process. So I have a filtration process, and then I will end up in different spatial complex. And then for each spatial complex, I can calculate the compilatory Laplacian matrix. And then I can check the change of the eigenvalues during the filtration process, right? So these are no surprising. If you check the multiplicity of the zero eigenvalue, it will go back to the Betti number. So it will be consistent with what you get from the persistent barcode. And then you can check the non-zero eigenvalues. And what you can see is that the non-zero eigenvalues clearly have much more fluctuations than the Betti number information, right? So this is no surprising because they are more sensitive to the structural change. So what we do is combine this topological information and the non-topological information together as features and then throw into the learning models. So the test case we use is this protein ligand binding affinity. The thing what we do is trying to predict how this ligand tend to bind to this drug. So essentially, if you have an efficient drug, what will happen is that it binds strongly with the protein that causes disease. So when they bind to the proteins, it will either activate or deactivate the proteins. At the same time, you want this ligand to not bind to other proteins that are irrelevant to a disease, right? So you have high specificities for this kind of things. So predicting the binding affinity is one of the most important steps in drug design. So here we check on some standard benchmark data set, and we found that our model works quite well. Another thing we try is the Ricci curvatures. Jürgen has already given a very nice talk about that. So the four-man curvature and Olio-Ricci curvature, they are quite different. Well, in our practical studies, we are interested in finding certain community and the linking regions. And what we found is that even though they are quite different, they somehow characterize the similar phenomena when it comes to community structures. So essentially, you see that here for this ORC and for this FRC, even though their values differ a lot, in the community regions, they all have positive values. And then in the linking regions or bridging regions between the communities, they usually have negative values. And this is actually what they used in the GM models to solve the over-scorching problems. Later, I will touch a little bit about that. And again, we are trying to not just checking the curvature for a specific spatial complex. Instead, I'm starting the change of the curvature properties during the filtration process. And then I collect all these things together, use as a feature, and then throw into the learning models. It actually also worked quite well. The reason that all these things works because mathematically they are highly connected to each other. So as mentioned, if you have the Hodge Laplacian, you do the decomposition, you can get the Betti number that goes to the persistent homology. And then on the other hand is that the combinatorial Laplacian can be related to the four-man Ricci culture through this Bochler decomposition. So essentially, the four-man Ricci cultures can be directly obtained from certain combination of the diagonal and off-diagonal component of the combinatorial Laplacian. So in this way, they are also strongly connected with each other. So far, what we're trying to do is that we have the data, we extract certain feature, and then we throw into the machine learning models to see how it works. But there's another more important thing we want to address is the representation, right? If we have the data, what can be the most suitable or what are the potential representations we can use? So again, in the biomolecular business, when people talk about molecular, this is the first thing come into their mind, right? It's almost like the golden standard of the molecular representation. So you have the atom, you have covalent bond between them, you have this covalent bond graph representation. So here, what we do is that we found that usually this covalent bond is in the region from 0 to 2 astrons. Okay, again, astron is 0.1 nanometers. So here in this representation, we just say, okay, any two atoms, if they are within the region from 2 to 4, I put an edge that. So I get rid of all this covalent bond. And then in this region, I collect the two atoms that are within the range from 4 to 6, 6 to 8, and 8 to infinity. So when I go to here, nobody would believe this is like a representation of molecular, right? You have zero sense of the common like of the molecular structures. However, even we use this kind of crazy representation, what we found is that based on 20 different benchmark data set for molecular properties, actually this crazy looking guy does not always give you extremely worse result. Okay, sometimes they can even get you some better result. Here, AUC, if it's larger, then it's better. You see here, this corresponds to this color, this guy corresponds to this guy. So you can see that in these properties is actually better than the covalent bond one. This one is also better. So what we're trying to argue here is not that this representation is better than this one. We are not trying to say that. But what I'm trying to argue is that the graph is just one way to represent the molecular. There are many different ways to represent that, right? It's not the golden standard. You have to use this representation when you study the things. Actually, if you read the paper of the geometric deep learning graph neural network, they use various different representations. Some people use this one, some people use k-nearest labels, some people use fully connected network. There are no unique standard way to do that. So in this way, can we go beyond to say use simplicial complex and hypergraph? That's what we're trying to say here. So other than the traditional, we are torus, Rips, check, alpha complex, we have trying to play a little bit with other different simplicial complex representations. So here, one model we played with is this neighborhood complex. It's actually from Lewar's work. So the idea is that when we have a graph, what you can do is build a simplicial complex directly from it. So the idea is that if I have a vertex here, I can find all the surrounding neighbors, and then I form a simplex among the surrounding neighbors. Here, for this guy, you have 3 neighbors. You can also form 2 simplex among the neighbors. So if you are familiar with the graph neural network, this is the things they do, right? They have the labels and then somehow aggregate the information from the labels, but here we assign it as the simplicial complex. And if you use this kind of neighborhood complex model on a bipartite graph, you will end up in something called a Dowker complex. Okay, so the idea is like this. For instance, if you study this protein ligand interaction, then I'm interested in the interaction between them. I don't care what are the interaction within the protein or what are the interaction within the ligand. I just want to see how one protein atom interacts with another ligand atom. So I end up in this bipartite representation. And then immediately you will see that all the labels for this green guy can only come from the blue guys. And then all the labels of the blue guys can only come from these green guys. So in this way, if I apply the neighborhood complex, I will immediately end up in two separate individual simplicial complexes. And what Dowker tells us is that these two simplexities, under certain conditions, they share the same homological information. So in this way, you can actually use one of the systems to explore the other systems. So this is the Dowker complex. And another thing we started with is this Hom-complex. I think Tina yesterday has given a very fantastic talk about motifs in the hypergraph. So essentially, people are interested to see what type of different motif you can find within the systems. Here, it's like you go a little bit further, say I have a structure, I have this K2, L3, K3, C4, K5, all this kind of motif. But now I want to see that how this motif type of structure form inner connections within the graph structures. So you have to do this kind of multi-homomorphisms. And this multi-homomorphism is kind of mapping from this motif into the structure. And then you collect all these mappings and then form a special polyhedron complex. So very roughly speaking, this is just like you have a certain kernel, and then you can somehow work this kernel on your graph, and then you get a new different representations. And this new representation will give you some extra information how these kind of motifs are connected in your systems. So this is about the Hom-complex. Another thing we do is with the hypergraph, more related to the talk here. So my collaborator Jie Wu actually proposed something called embedded homology for hypergraph. So essentially, when you have a hypergraph, you do the boundary operator, usually you will have some trouble as it may not always be consistent. So what you can do is that you go from the hyper edges into the chain level, and then in the chain level, you can try to do two types of operations. One is called a superior chain complex and inferior chain complex. The general idea is like this. You have this chain component from the hypergraph, and then you can try to add in component into it so that it will be consistent under the boundary operator. So this is called a superior chain complex. And then in the inferior chain complex, essentially is that I get rid of the part. When I do boundary operator, it will have trouble, right? So you get rid of this part. You only preserve the part that under the boundary operator, it will be consistent. And what you found is that the homology defined from the superior chain complex and the inferior chain complex will be exactly the same, isomorphic to each other. So we use this idea, the embedded homology, to study the interactions. So again, we try to define some special hypergraph based on this bipartite interaction, and then we extract the homological informations and then use it as a way to study the systems. The very last thing I want to mention about this representation is the Tor-algebra. So here we try to jump a little bit outside the simplex representation. So you have simplex, but now you can define monomial on each of the simplexes. And then you collect all these simplex monomial together to define something called a Stanley-Razor ring structure. So when you have this ring structure, you can study the Tor-algebra of this one. And this Tor-algebra can be decomposed into this bi-gradient component, and each of these bi-gradient component can be studied through the co-homology. Okay, it looks a little bit complicated, but what these guys really do computationally is like this. So you have a simplicial complex. Previously, I just have the chain complex, I calculate a homology from it, right? But in the Tor-algebra setting, what you really do is check the homology, the co-homology of all the four subcomplexes. So that means that you can do this way. I take any two atoms from this system, and then I find all the four subcomplexes formed by these two atoms. And then I can take 3 atoms from this system and all the 4 subcomplexes from these 3 atoms, so on and so forth. So in this way, when you collect all this homological information from all the possible 4 subcomplexes, it will give you the Tor-algebra information. So it's much richer information than the common persistent homology. And the beautiful thing is that this Tor-algebra, you can totally bypass the computation, the persistent homology calculation using the, some computational algebra softwares. So this provides another more algebraic representations, and all these representations we can use it to study protein-protein interaction. Recently, we also do some work in the material design. But more interesting, what we want to do is build on these new representations to get into the geometric deep learning and topological deep learning the things. So again, in the geometric deep learning, there's a lot of fantastic works, and yesterday Patricia has already given a very beautiful lecture about that. So one thing we started with is that we're trying to say the curvature, how it's related to this over scorching information, and we found that this geometrical information indeed works quite well. However, in the real molecular structures, you seldomly see this kind of linking regions. Because you can imagine if I have two clusters, this linking region in between, it will be extremely unstable for the real moleculars. So in the real moleculars, what we're trying to see is to study the local type of information. So here, what we consider is the randomized torsion or analytical torsion. Very roughly speaking, what this one really does is that it characterizes the complexity of the structure by studying how this structure should be embedded into a certain space. So imagine you have a torus, you have the Klein bottle, right? The torus, you can embed it in R3, but the Klein bottle, you can never embed it. You need a higher dimensional structure for you to embed this torus, this Klein bottle into. So this analytical torsion roughly tells you how complicated your structure is by checking the potential complexity of the embedding space. So what we do is that use a certain special version proposed by Yao in 2022, and then combined with the message passing mechanisms. So essentially, locally, we input this information into the learning systems so that it can be aware of the local complexities. So this is one thing, but we really want to do is the topological deep learning. So here is that we have structure, we try to see what can be the suitable representation. Because the data we study is not really that large, so we just do a filtration process, and then we pick up several structures and different scale we believe has the chemical importance. And then for each simplicial complex, we can do the message passing at different level. So you can have load level message passing, edge level message passing, and simplicial complex message, two simplex level message passing, and then collect the information together. So essentially, is that you have the message passing or the information exchanging at a different level, and then collect them together. And recently, what we did is using this kind of ideas to study the material data. So the material data, when you view it from the molecular level, is actually quite different from protein DNAs. This is because they have very interesting periodical information, like you see here and here. So essentially, what's going on is that you will usually have a certain structure plus a unit cell structure, okay? This unit cell is basically like a periodical condition. So you can imagine you have one structure plus a certain periodical conditions. If you change the periodical conditions, even though your structure can be the same, when you goes out, laid out, it will be dramatically different. So the idea is how we incorporate this periodical information into the setting of the molecular structures. So previously, what people did is this Curvature graph. So in the Curvature graph, what they done is that they have a certain graph representation, and then they identify the atom of the same type, and then they put all these atoms together. However, during this process, they don't, make these edges in the same way. So in this way, they end up in a graph, a multi-edged graph. Okay, so what we do is generalize these things into the Curvature complex. So essentially, we can also glue the atom together, and then we can have this kind of cellular complex between them. So in the real calculation, the business is like this. So you have a unit cell, and then what they can do is repeat it for many, many times. So x-axis is 3 times, y-axis 3 times, z-axis 3 times. Then you end up with something called a supercell. So this supercell has many individual unit cells in it. And then they convert this entire supercell into a multi-edged graph. So in this multi-edged graph, you have edge information that represent intercellular interaction. You also have edge information come from extracellular interaction. Okay, so what we did is that replace this in with the convolution complex, and then bring into the settings. We design some transformer structures to consider this kind of information, and then throw into the the learning process. And we found that it actually can work slightly better. And another thing, what we try to do is take some physical insight into the black box. So now you have a lot of deep learning models that you can have very fancy architectures, but they boil down that people are always worried what are the missing components, right? How you preserve symmetry, how you preserve certain conservation law, how you preserve certain physical insight. So here, when we build up the architectures, we try to mimic what the molecular dynamic does. So in the MD force field, what you will see is that you have something called a bound energy. So this is related to the covalent bond and a certain non-covalent bond. And then you have something called a bound angle energy. So this one is related only to the covalent bond. And then you have something called dihedral energy. So this is related to that you have 4 atoms, each 3 of them form a triangle, form a plane. This one have another plane, and then these 2 planes you have an angle, and this one contribute a significant, a very important term in the force field. So when we are building up our simplicial complex, we are trying to mimic each of the term in the molecular dynamic force field. So in this way, we can build up our special path complex, and then we can learn the features. So again, we have to do some iterations and make sure the information can be passing between different type of atoms. And then we try to collect the information together and to see how it works. So to quickly summarize what we want to do in general is to combine the mathematical tools from topology, geometry, and algebra into the black box of the AI models, right? Make them aware of the symmetry, make them aware of some of the physical insight, aware of the geometric informations, and then trying to see if you can build up some more efficient models. And this is my group and my funding agencies. I will stop here. Yeah, thank you.
Great, thanks, Colin. Do you have any questions?
Yeah, you now have a very rich spectrum of methods that you all master and that you can all apply. But when you get particular biological data, we have some intuition which kind of method would work best so that you don't have to try all of them.
I think we can start it from TDA. So that's my general understanding, because the structure connectivity you usually think is very important, and the topology characterize the connection, the informations within the system, and that may be one starting point for people to try. Yeah.
I have a question. Like if you were a deep learning evangelist, they would say, you don't need to bother with all this stuff. Just have the right model and you can learn all of these features from the data. So how would you respond to that? Is it to improve the performance of the model? Is it for like interpretability? Does it make better use of data? What's the sort of core advantages of combining the mathematical insight and topology alongside the machine learning?
I agree. You see, in the maybe 5 years or 10 years ago, people are saying, okay, I have a lot of money, so I can, I collect a lot of data and then buy the, 800 GPU card and I can get the business done. But nowadays, I think people begin to more aware of the importance of the, preserving some intrinsic information, so like geometric deep learning. There were, Pel gave a very nice talk yesterday. He already mentioned that you need to preserve certain, translation invariant, this kind of different invariant informations and, particular equivariant informations when you study the force type of stuff. And another thing is that, the whole black box type of deep learning, still a lot of people are worried about that, how to make it less black, make it more transparent, make the physical and mathematical insight into it. I think now many CS person are beginning to change the mind to say that the brutal force way of doing the things can get everything done. It's not only data driven, but maybe guided by physics, guided by mathematical principles. So in this way, I believe, especially when they trying to dealing with real chemical data, there's a lot of the symmetry information, energy information they have to preserve. Otherwise, they can use diffusion models, they generate some structure that may totally does not represent the real data. They have to preserve certain important properties, and these are the part opens great opportunity for mathematicians to play the game.